Never date a geneticist. They'll always find flaws in your sequence.
In your sequence? Welcome to Absolute Genius, a podcast series from Thermo Fisher Scientific. I'm Lisa Crawford.
And I'm Jordan Ruggieri. We're excited to welcome today's genius, Thermo Fisher's very own Dr. Kathie Sollweck.
Kathie has been with Thermo Fisher since 2022 and currently serves as a senior medical affairs manager working with oncology. We loved learning more about innovations at the forefront of oncology research and how Thermo Fisher technology is advancing the field. Thanks for joining us, and we hope you enjoy our conversation.
Kathie, thank you so much for joining us today on our episode of Absolute Genius. Really, really excited to have you. Could you maybe give us a little bit of background and what you work on and where you work?
Yeah, my name's Kathie. I work as a medical affairs manager for oncology at Thermo Fisher. Yeah, I used to start as a sales rep actually in Thermo Fisher, and I sold the Absolute Q for a while, and I was a technical specialist for digital PCR before moving over to medical affairs, and that's where I'm at.
That's awesome. And working with oncology seems like a really exciting field. Can you talk a little bit about your involvement in research with customers or scientists around oncology?
Yeah, I started out with oncology right after my PhD, worked in a clinical lab, and that's kind of where I actually had my first contact with oncology and all that surrounds it. And yeah, that got really close to my heart and sparked my interest, let's put it this way. When I moved to Thermo Fisher, I was kind of naturally drawn to those customers just because they fascinate me, they interest me. I feel they're doing such important work. And so from the very start, I was very close to them, very close to our pathology, oncology, hemato-oncology labs, but also anything microbiology or just in the, let's say, oncology space.
Absolutely incredible. Really, really, really interesting to hear. From your perspective, what are some of the most pressing unmet needs in oncology research, or monitoring, or really anything of scientists in the field today?
When we're talking about unmet needs, we really want to make it as easy as possible for the affected people. So, what research is kind of focusing on is how can we have the biggest impact for anyone out there who could be affected by any type of cancer and without, having any compromise on therapy. So, what is really needed is easy methods to access these cells, basically these tumors, and then easy methods to classify them and identify therapy selections that we can use for these people, essentially. So, obviously, the field of molecular oncology has been moving a lot, and that brings a lot of benefit to the field since we can genetically classify a tumor, and then that will give us a lot of information about how we can respond to that. And I think what is mostly needed is easy access. So there's a lot of research about, liquid biopsy, blood, or identification from urine. Also, stuff like fine needle aspirates, or all these things that tend to be easier to access than a big surgery and tend to be also easier to classify the tumor of some sort.
You talk a lot about access and liquid biopsy. What are liquid biopsies?
A liquid biopsy can be blood, it can be cell-free DNA, it can be circulating tumor cells. It can also be urine or ascites, or even like brain fluid. So a liquid biopsy just means we're not taking a solid piece of a chunk of cells, but we're taking something liquid that is usually a lot easier to access from the body and therefore a lot easier to access also for the people who are affected. If you have to come in for surgery every time you want to see what happens, if there was a therapy response or not. That's obviously a lot more intense and painful, and also these people might not be in the best state when they have to come in. So it's definitely a lot easier to look at anything liquid. And then obviously from these entities, there's different types of analyses that you can do to kind of observe what is going on, what happened, or where we have to go.
Are there certain kind of cancers that lend themselves better to liquid biopsy testing, or is that still kind of under study, or can you use it for most types of cancer?
So that's a big, big study field. So far, liquid biopsy is very common for lung cancer. We've been doing that for, I don't know, at least 5, 10 years to follow therapy resistance in lung cancer in liquid biopsy. Now um, the newest kind of stories that we follow in liquid biopsy is breast cancer, and we're moving ahead there a lot. And yes, we can definitely classify cancers in shedders and non-shedders. So cancers that shed DNA to the blood or anywhere else a lot, or cancers that don't really do that. But we also have to be aware that we always have cell-free DNA, for example. So there is always a baseline, and that baseline also can tell us a lot about the health status of a person.
That's amazing. I think it lends directly into my question was, can you talk a little bit about some of the challenges of using liquid biopsies? I mean, I can imagine if you're taking from blood, for example, or taking a blood sample, that there's a lot of stuff in there, right? Is it easy to detect some of the shed DNA or circulating DNA, or is it quite difficult?
Actually, I wouldn't say it's difficult. The challenge that we have is the quantity that's in there. Obviously, there's not much. And that's again where digital PCR also comes into play. We need methods that are highly sensitive to be able to analyze this type of DNA. Because imagine your breast cancer sheds a lot. Majority of that DNA will still be a normal, healthy, wild type DNA. So even though you might have okay amounts of DNA in there, I don't know, 20, 40 nanograms, your mutational burden and is usually pretty low. So you still have to find a method that is able to detect those low level of mutational burden. So that is definitely a challenge. And also we have to be aware that with anything liquid, we have to be very stringent with pre-analytics. So we have to make sure that the general amount that we find is actually cell-free DNA and not some, blasted blood cells that shed all of their genomic DNA in there.
I actually want to talk a little bit about some of the workflows and technologies utilized. And you mentioned a couple even in our conversation so far with digital PCR and NGS and sequencing. Where do those two methods? I mean, we love DPCR, we love sequencing here at Absolute Genius. Where do they fit in those two when it comes to oncology workflows?
Well, we can look at NGS as kind of our wide angle lens. We use NGS to look at thousands and thousands of genes and kind of get a glimpse of the situation that we have. So we use that to genetically classify our tumor. And we might use NGS for identifying certain types of mutations that either cause a certain type of pathway to stop or maybe even enhance a certain other pathway, something that might lead to resistance against certain therapies, but also to identify new biomarkers, for example, that might be needed for future clinical trials, anything like that. And then we have digital PCR that is kind of like the laser pointer. Like once we identified a mutation or an alteration that is important for us, we have now digital PCR to follow that up with a very high precision and a very high sensitivity. So we will use digital PCR to monitor the disease over time, to look at progression or at relapse, but also to look at resistance mechanisms that might come with a certain sort of therapy. And that's how these two methods work very well with each other.
You're seeing like NGS would be more along the discovery, right? You're trying to find certain biomarkers or you're looking at a whole sequence to help classify versus digital PCR is more along the like quantification element. Is that correct?
Yeah, both of that, but also you can quantify with NGS, just not at these low levels. So let's say we take NGS and I would say the most precise NGS methods can maybe achieve a sensitivity of 0.1% mutational burden. And that means that I have, 99.9% wild type and 0.1% of my mutation. Whereas with digital PCR, it can go down a lot further. So yes, NGS can give us also a quantitative result of some sort, but not in those very, very low abundance regions that we often have if we have to monitor or assess resistance and all of this.
Ah, okay, makes sense. How do researchers decide when they need to move from like NGS into a digital PCR? Is there a decision point that comes from there, or I don't know? Is there a threshold on NGS where it's actually I might need to use digital PCR to help get more accurate?
So, well, the first questions that we have here is definitely what is it that I want to do? Obviously, NGS can be used for most of the things, but it's also very expensive. We have to think about that as well, also in research. That doing an NGS, if I'm actually only interested in two, three mutations that might cost and affect something, is absolute overkill financially, but also from the workload. An NGS, it does take overnight at least, whereas the digital PCR, you can do that in 60, 90 minutes if you want it. Um, so a real cutoff, we have to differentiate a little bit of the fields that we're looking at. So if we say we look at solid tumors, for example, then there is not really a cutoff that we need to achieve. There it is more a practicability of things. Let's say I find some sort of mutation that I need to follow up. There's no need to do the whole NGS every time. If I look at liquid biopsy, once I identify something in my tissue and I want to see the liquid biopsy of it, I will move to digital PCR just to be more confident in the precision and in the sensitivity. If we look at the hematological space, anything leukemia, we will always need the NGS because those kind of blood cancers, they are so heterogeneous. There's so much going on that in this first instance, we will never think twice. If we do a targeted method, we won't do that. It's impossible. But then once we found the culprit, kind of, we can go and use also that more sensitive method to follow up on how the disease is progressing or if the therapy is working or not.
Is there a particular workflow or a particular order that these are used in NGS, dPCR, qPCR when a researcher is looking at a sequence, validating they have the right sequence, the right primers, probes in order to move from an NGS into something like digital PCR?
That's a tricky question because again, that is very heterogeneous. Okay, so that really depends on the cancer type we're talking about. So I think a very good example for this again is hematological diseases, because here what we do is identify something with NGS, and that can be very person specific. Here, what we do is we will find something with NGS. And then what we'll do is we'll design primers and probes for that exact thing that we found to be able to follow that up. And that might not only be one, that can be up to 15 different things that we follow up. Then there's other cases where, let's say, there's only a specific set of activating mutations that are activating a certain pathway and that are interesting in that segment. And in that segment, you wouldn't need to kind of verify, do I have the right primers and probes? Because I do. But when I found it in NGS, I can pick and choose the right one and follow that one up. And if we're saying we need to cross check certain things, yes, we do. But that wouldn't be done with the PCR method. We have, for example, in NGS, there's things that are tricky, which are like large deletions, large insertions, or if we have a ton of repeats of the same thing. And then you would actually come in, for example, with the Sanger sequencing methods to kind of check, okay, this looks weird, it looks off, like the copy number might be weird, or I might see certain drop-offs. And then I might confirm that with an orthogonal method. That would not be, though, digital or qPCR. That would be something different. So I don't think we need to cross check with the digital and the qPCR and the NGS because essentially, if we think about it, originally it's the same thing. It's all the PCR, and we're kind of either looking at every single base that's in there, or we're looking at a specific kind of point mutation or exchange with a probe.
Makes sense. That's what I was looking for, was that tie some of that tie in with the Sanger sequencing. So awesome. I just didn't it didn't cross my mind it was Sanger sequencing, but yes, makes total sense. Um Kathy, you talk a lot about MRD. Can you elaborate? What is MRD?
MRD is minimal residual disease. It is, again, especially important in hematological diseases, but that is also a very fast moving field. Now, there is also big interest for MRD in solid tumors. That is coming more and more, and MRD can look very different. MRD can be I look at very low level amounts of baseline mutations that I find, or I look at methylation patterns of a tumor, and that methylation pattern might indicate to what level I have residual disease in there. Let's say the most classical description of MRD would be the hematological one, where we have to be super, super stringent in following up during therapy or, let's say, after a transplant, because if we have one single cell left in there that carries that specific activating mutation or a fusion, whatever it is, that one single clone, if we miss it, the whole circle starts again. So that is the very original description of MRD. We want to make sure we find every single cell or clone in there that still carries the disease-making variant or whatever that is.
That's really, really interesting. Kathy, a lot of things you're talking about as well, it seems very personalized, very, very specific to a subject or to the research or to an individual. How are scientists and researchers managing this data and managing some of these workflows as they come in?
Yeah, so obviously we have a big move also direction AI and everything connected to that. We have to mention that for sure. So there's some labs, they don't even do the microscopy like under the microscope anymore. They just have digital slides, or doctors, they used to dictate into a microphone what they're seeing under the microscope, and that is also, take AI is taking that over slowly. But obviously, if we speak about the molecular data, we also speak a lot of whole genome sequencing, whole exome sequencing. Biggest challenge here is the storage, because obviously you have to store that data for a long time. And so every institute has to find solutions, servers, clouds. And then also that depends a lot on the bureaucracy of that specific country. So it's really, really hard to save and store, data like this, big amounts of data. So definitely that is a challenge. And that is also one of the big questions of the field. How are we going to handle this type of data, since it's only going to be more of that in the future?
Especially, it's not like it's one size fits all, right? If you're sequencing every single sample that comes in, man, that's a lot. What drives adoption of new technologies in oncology research? Is it very heavy on kind of the published validation and kind of the published findings? Is it more internal pilots? Is there anything that really drives adoption?
In my own point of view, in my opinion, adoption in daily life is mostly driven by new therapies that are coming out. So the pharma industry is kind of largely deciding with the therapies they get approved, what is going to be tested and how. Because obviously, for a healthcare system, it all has to make financial sense as well. So, if I have a drug that's being approved and it's really working very well, obviously that will be prioritized in adoption for testing. Let's put it this way. But also now, many countries have like more specialized cancer centers called centers of personalized medicine, for example. And these centers, they are able to also implement testing methods, therapies that are only, from clinical trials maybe, or also just research that has been done. So there is possibilities if someone was to need something that is not in the approvals and not in daily life yet. Let's put it this way.
Are you looking to precisely quantify rare targets in your sample? Are you always struggling with standard curves or measuring limits of quantification? If so, Applied Biosystems QuantStudio Absolute Q dPCR system is for you.
With the Absolute Q, reliable and precise digital PCR can be as simple as preparing your samples, loading them onto the plate, and running the instrument. The system helps consistently generate more than 20,000 microreactions with a low percent CV and provides results in as little as 90 minutes.
That's right, Lisa. And the Absolute Q utilizes more than 95% of the sample input. So you won't have to worry about missing rare targets due to dead volume. You can learn more at thermofisher.com/absoluteq or visit the Absolute Genius webpage. The Absolute Q is for research use only, not for use in diagnostic procedures.
Okay, let's go back to our guest.
All right, it is time for the career corner of the episode. Passing it over to you, Lisa.
Thanks, Jordan. So, for the Career Corner, what we like to do is talk a little bit more about you personally and how you got to where you are today in your career. Did you always want to be a scientist?
No, actually, no, I did not. I wanted to be an actress for most of my young years. So, no, not at all. That changed a lot then, I would say, in my teenage years. I wanted to be a vet, actually, for most of that time as well. And then, for some weird reason, I was into plants a lot and I wanted to become a botanist. And that's why I actually started studying biology, which is wild seeing where I ended up now. I have to also, as a disclaimer, I have to say I'm a very go with the flow person. I am not someone who is a planner. I am not like saying I want to be there and there in like 5 to 10 years. Not at all. So I'm much of a no pressure, let's see where this goes kind of person.
Especially for science, I feel like there's a lot of you kind of have to follow where it takes you as opposed to you dictating where it goes. Was there a moment you remember that kind of switched on your interest specifically into oncology and kind of led you into your higher education and your PhD?
So I have to say my PhD has nothing to do with oncology. So my PhD is in bioanalytics. I actually looked at antibiotic resistance in water, and that was already very industry tied. So I actually always knew I did not want to stay in academia. So that is why I chose a PhD that was very real world industry related. And the way I ended up in oncology was actually just after my PhD. It was the first job I applied to, and it was the first interview I had, and also that was successful. That is kind of how I ended up in oncology. So I didn't even know until after my PhD that I would like that so much. I thought I was more of an still like medically related, all the pathogen and antibiotic resistance, and but I thought I was going to be more of an like environment kind of person. But sometimes you just have to do things to see that you like them.
Well, I always think that means that this is where you're meant to be because something intervened and pushed you onto this track. Have you had any mentors that you've interacted with that have really helped shape your career and your interest in your path?
Yes, definitely. But I would say most of the mentors I'm super grateful for I had within Thermo Fisher. So when I moved from the clinical lab into Thermo Fisher, I discovered a whole new world of very, very supportive people. And I also have to say I owe these people everything that I have done ever since I'm with the company. I've had wonderful mentors. Usually my colleagues, I had one colleague in particular, she brought me into Thermo. She was my mentor for my first year. And then after that, I had my managers. And also, we usually in Thermo, we always have to choose a mentor. So we have the mentoring program. And that is also a really good thing to do because you have someone who pushes you to reflect on yourself and where you want to go and what you want to do, what your passions are, and all of that.
Now that, you've achieved what you have in your career, do you have advice for other people who may be wondering about your path as an option? Because I know, a lot of researchers, oncology, they go into academic research, like you said, that wasn't your path. But any advice that you would have for someone interested in kind of going the same direction you had and maybe going a little bit more towards, the company side of things as opposed to academic?
So the first thing I want to say, don't be scared of being a salesperson. Sales is often an entry point into companies. And it has a really bad stigma somehow. And I do not understand why. It's the best job in the world, really. I did not leave sales because I wanted to leave sales. It was just a natural next step that evolved and really interested me. If you are a sales rep or if you're a technical sales specialist, you are at the forefront of all these scientists. You support them, you provide them with the best technologies that they can use. You can be super interested in their projects. You can collaborate with them. That's the first thing I would like to say because many people, when they hear that I started as a sales rep, they're like, "Oh God, no, I could never." I'm pretty sure you can. And also, it makes you grow so much. You have to speak to strangers every day. You have to make connections. You have to talk to people who are probably way smarter than you are, at least in the beginning. And that is my first step of advice. And also, secondly, you have to start somewhere. I don't think the place where you enter a company or the corporate world is the place where you're going to end up. It will never be. So, even if that job that you have offered there right now is not your dream job, just, you have to start and open the door and meet the people within there and prove yourself somehow. And from there on, you can definitely also develop in different roles. I did never think I was going to end up in medical affairs. 1.5 years ago from now, I did not know that existed. So, is this how things evolve? Yes.
It's always interesting to see. I mean, I love the advice as well, but I think what people don't understand is, especially when you're going into industry, you as a salesperson, you're talking to scientists, so you still need to know the science, and you're advising them on what challenges they're having and what technologies might be able to help. So I think a lot of people who are in the lab, they think going to a sales role is being removed from science, right, and from research, but it's not the case. Is that also your experience?
That and also kind of if your customers are scientists, if you don't know anything, they'll not take you seriously. So you have to actually know your stuff. Otherwise, you're lost. And also, secondly, it also has the stigma of, well, if you just show up and talk to people, they're just going to be annoyed. I don't think there ever was an annoyed person during my time in sales. There was nobody being like, oh, close the door or go away or never. I think that is also like a prejudice that you have. Usually people are really grateful to have someone they can talk to. Anything that comes up for them, I need a new extraction kit because I have these weird cells now. Who do I ask? And it's good if you're the person they think of. And that's also really exciting because you're gonna also have to think about which extraction kit do I give these people now. So there's a lot of thinking involved. Obviously, also a lot of numbers, a lot of quoting, a lot of pricing, but also that I didn't know any of that when I came to the company. That's a lot of good things to learn too, business wise and how to make price models, how to decide on pricing, how to negotiate for that matter. Those are all good life skills that I don't think don't hurt anyone.
Yeah, I think sales is kind of a four-letter word, I guess, especially culturally in the United States too, because you have, no offense to car salesmen, but, that's kind of where everyone's mind goes. But you can kind of reframe it as you're less of a salesperson and more of a bridge between the scientist and our company and really just helping them get what they need and accomplish what they need rather than giving them things they don't need, which is why I think a lot of people associate negative things with sales. So, but no, even you talking about it, I feel better about salespeople already. Just a fun little question for you to, kind of wrap this up here is do you have a most embarrassing moment that you maybe have experienced in, your career journey and then a proud moment just to balance it out?
Well, I think the most embarrassing moment, I met a very important customer once, and he has kind of a funny last name. I don't want to mention it here. But I was like, no, that's not your name. And I was kind of laughing because I thought they were joking. But they were not joking. And that was a very important guy. So, yeah, that was probably my most embarrassing moment as that was already within my role now. So, supposed to be a very serious person. So, no. Most proud moment was probably during my last year when I was a technical specialist for the digital PCR because we, together with one of our most important customers, we managed to build a whole kind of testing pipeline that was adopted throughout all the territory. Lots of people adopted that for a new drug that was launched. And so I feel very proud of that, to be able to build something like this from scratch, just with the help of our beloved customers who supported that. Yes.
That's awesome. Really cool.
This has been a really fascinating conversation. I guess a question would be, because we don't have hours and hours to go into everything with you as much as we would like to, is there anywhere that someone who's interested in learning a little bit more about the sort of things you talked about can visit and get some more information and do their own research?
Of course, there's a lot of resources. You can look things up. There is a really great resource that I like a lot. It's called OncoWeb, because it has everything. It basically has for every type of cancer. It has biomarkers. It has which mutations fusions are important. It has therapy relevant things on there as well. But obviously, we also have Thermo Fisher resources, like our oncology research website that has all the methods on there as well. So if you're ever wondering, how does one extract cell-free DNA, or how do I do pre-analytics, or what instrument can I do fragment analysis with, and which one does the Sanger sequencing, or what's the portfolio of NGS I can use, or what is my assays I can use in digital PCR, they would all be there. You can click through the single methods on there.
That's awesome. I know those questions keep me up every single night. So I will make sure for our listeners as well that we put those in the show notes and that everyone can have access to those and read through them and hopefully learn a little bit more.
Kathie, thank you so much for joining us on today's episode of Absolute Genius. Really, really interesting stuff, and really glad that we were able to talk to you.
Thank you so much for having me. I hope it's not too messy. It might be a little messy, right?
All good.
That was Kathie Sollweck, Senior Medical Affairs Manager at Thermo Fisher Scientific. This episode of Absolute Genius was produced by Sarah Briganti, Matt Ferris, and Matthew Stock, with more great conversations around the corner in upcoming episodes. Stay curious, and we'll see you next time. If you're happy, I'm happy.
I don't know. I tried to make a joke about transcription, but it didn't translate well.
Lord. Okay, I'm done, so we can make him stop now.